Swayam Prabha. Digital India. Educated India.
So students, this completes the process of spermatogenesis. Here we have discussed the three phases of the spermatogenesis in detail. The multiplicative or the proliferative phase, in which the sperm gonial cells increase in number by extensively undergoing mitosis. Then the meiotic phase, during which the haploid spermatids are produced. And finally the spermiogenesis, during which the spermatids produce the functionally active and motile male gamete known as spermatozoa. We also discussed the structure of the sperm in detail, and finally we have discussed the functions of the sperm. Now we will start the process of oogenesis. So what is oogenesis? Oogenesis is the process of formation of mature female gamete, that is ovum, in the ovary. Now, just like the primordial germ cells reach the embryonic testicles, similarly in the female embryo, the primordial germ cells reach the embryonic ovary. Now, the primordial germ cells do not originate in the ovary. They originate rather in the posterior portion of the female embryo. And then they cross the dorsal mesentery to migrate into the gonads via hindgut. And this happens by the 6th week of gestation. Now, this migration is controlled by various factors, predominantly the bone morphogenetic proteins and activins, both of which are a part of the TGF-beta superfamily. So, students, just like spermatogenesis, oogenesis is also completed in three phases, namely the proliferative phase or the multiplicative phase, during which the oogonial stem cells will increase their number. Second is the meiotic phase, during which the reduction and division will take place. And third is the maturation phase, which involves finally processing the ovum and making it fertile, ready for fertilization. So the first phase to start here, multiplication or the proliferative phase. Now, during the multiplication or proliferative phase, which happens before birth, now this is the major difference between the proliferative phase of spermatogenesis and oogenesis, that in spermatogenesis, the multiplication phase goes on even after birth. But in oogenesis, the process of proliferation ends before birth. That is, the number of oogonia or the germ cells increases only in the embryo, not after birth. Right? So in this phase, the primordial germ cells undergo repeated mitotic divisions to produce undifferentiated germ cells called oogonia, which are diploid in nature. Third month of gestation in the female embryo. Now, after repeating mitotic divisions, these become the primary oocytes by 7th month of gestation. Then these primary oocytes start the process of meiosis, but they remain arrested in the diplotein of prophase 1 of meiosis, and this is known as the dictate stage. At this stage, the primary oocytes are surrounded by a layer of follicular cells, and thus they become a primordial follicle, as is shown in the diagram here. You can see this is a primordial follicle in which the primary oocytes, which are arrested in the diplotein stage, they are surrounded by follicular cells. They have certain thecal cells around themselves, and they're the granulosa cells around each of the follicle. And this complete is known as primordial follicle. So we can say the primary oocytes remain inside a primordial follicle during the multiplication phase. And these primary oocytes remain arrested in prophase 1 diplotein stage till the puberty is reached and they start undergoing the process of growth. Now the second phase is the growth phase, and this phase continues till puberty. At puberty, in each month, alternately in each of the ovary. Some 15 to 20 primary oocytes or the follicles, they begin to mature. But each of them does not mature completely. Usually only one reaches full maturity in humans, as we say, and rest undergo degeneration or atresia. And such follicles are known as atretic follicles. Now, as the primary oocyte begins to grow, it gets almost 100-fold increase in its mass. Now, the surrounding follicular cells proliferate to produce a stratified epithelium of granulosa cells, and this unit is now known as the primary follicle, as you see in the diagram. The nucleus of this primary oocyte becomes very large due to increase in nucleoplasm and is now known as germinal vesicle. This long growth phase gets completed in two subphases. The PREVITELLOGENESIS, in which large changes happen to the cell organelles, and VITELLOGENESIS, when yolk is laid down. So we can say during growth phase, a primary oocyte is enclosed in a primary follicle. As you see in the diagram here, which shows the number of germ cells in the human ovary changing over the lifespan, we see that the total number of primary oocytes at birth varies from 2 to 7 million. Like before birth, there is extensive proliferation of the germ cells, which shoots up to almost 7 million. Some of them undergo atresia, so that at birth, some 2 million are shown here in the graph. Now, during childhood, most oocytes further undergo atresia, so that only approximately 400,000 are present at the beginning of puberty, and even fewer than 500 will be ovulated in the entire lifespan in humans. Now, as development continues, the follicle cells secrete a follicular fluid or the liquid folliculi in the intercellular spaces of granulosa cells, which are known as antral follicles. Eventually, these antral follicles fuse to form a large fluid-filled follicular cavity known as antrum, and the follicle is now known as secondary follicle or the Graafian follicle. Now further, the granulosa cells around the oocyte secrete a layer of glycoproteins on the surface of the oocyte, forming the zona pellucida layer, which is a non-cellular primary membrane. Zona pellucida and a few follicle cells will persist when the egg is ovulated. This zona pellucida is primarily involved in species-specific recognition of the sperm, which is helpful during fertilization. Now, zona pellucida further comes to be covered by another layer known as corona radiata. The few layers of corona radiata which remain intact around the oocyte form the discus proligerus, which remains attached to the follicular wall by a stalk of follicular cells called the cumulus oophorus or germ hill. Now, this developing oocyte is further surrounded by all the membranes, then granulosa cells, and ultimately this Graafian follicle is surrounded by a large number of thecal cells in layers. There is also a layer of cortical granules formed under the oocyte plasma membrane in some species. Now students, this Graafian follicle appears bulging on the free surface of ovary. And this free surface weakens and stretches to produce a specific point known as stigma. This stigma is actually the point of rupture of the ovum when ovulation occurs. Now in this diagram, they have shown the oocytes developing in the follicles located in the ovary. At the beginning of the menstrual cycle, as you see, the follicles are maturing. At ovulation, the follicle ruptures, releasing the egg. The follicle thus becomes a corpus luteum, which eventually degenerates. The B diagram is showing a follicle under a light microscope which has got an oocyte at its center. The third phase is maturation phase. So maturation phase of oogenesis involve all the processes that prepare the ovum for fertilization. During maturation process, a surge in luteinizing hormone stimulates maturation of the ovum and also ovulation. There is extensive chromosome condensation taking place during maturation phase. There is also a characteristic feature of maturation phase known as germinal vesicle breakdown, which involves disintegration of nuclear envelope. During maturation phase, the meiosis 1, which was earlier arrested at the diploid phase, is completed, as you can see in this diagram. This results in the formation of two daughter cells but of unequal size. See, the cell which is larger in size, which receives most of the cytoplasm, is known as the secondary oocyte here, while the other is known as the polar body or the oocyte. Here it is known as the first polar body, and this daughter cell receives none of the cytoplasm. Now this secondary oocyte will enter meiosis 2, but is again arrested in metaphase 2, approximately 3 hours before ovulation. Now this meiosis 2 is completed only and only if the oocyte is fertilized by a sperm. Otherwise, this cell will degenerate approximately 24 hours after ovulation. So if meiosis 2 happens, it generates a mature ovum, and there is also production of another polar body. Now these polar bodies have very little cytoplasm left in them. So they disintegrate or remain inactive, or simply they again divide into two polar bodies in different species. And the nucleus of the mature ovum that is produced after meiosis 2 is known as female pronucleus. So student, one thing important to note here is that meiosis 2 is arrested before fertilization by certain factors, important of which is cytostatic factor. But during fertilization, there is heavy influx of calcium ions, which inactivates the cytostatic factor and thus helps in resuming meiosis 2. So we can say that the secondary oocyte, which is ovulated from the ovary, arrives in the oviduct for fertilization when it is present in the metaphase 2 stage. The meiosis of a secondary oocyte is completed only if a sperm succeeds in penetrating its barriers, otherwise it is degenerated in this stage only. So if meiosis 2 resumes, it produces the exact haploid ovum, which is just at the entrance of, or which is just going to enter fertilization. So we can say that ovum is just a brief transitional haploid stage between the diploid oocyte and the diploid zygote. So what happens to the Graafian follicle after it releases the ovum? After ovulation, the Graafian follicle degenerates in the form of corpus luteum. Now this corpus luteum retains some of the thecal cells and some of the granulosa cells. If fertilization happens, this corpus luteum becomes large, glandular, is sustained for some time during pregnancy, and it produces progesterone necessary for implantation. If fertilization does not occur, then corpus luteum degenerates to form a haline structure known as corpus albicans. As discussed during the growth phase of follicular genesis, that the growth phase of primary oocyte gets completed in two stages, PREVITELLOGENESIS and VITELLOGENESIS. The PREVITELLOGENESIS involves no yolk formation. That means it is the phase which takes place before vitellogenesis, before the formation of yolk. So it majorly includes the changes in cell organelles. So there are changes in the nucleus. Nucleus becomes very large due to increase in nucleoplasm and is now called the germinal vesicle. The nucleoli also increase in number and size due to increase in transcription of rRNA by the rDNA of the nucleolar organizer region of the chromosomes. Now, important to note here is that not nucleoli of all the organisms increase in number. Some have very high number, some have quite low number of nucleoli in them. For example, the primary oocyte of sponges and echinoderms have just one large active nucleolus present in them, whereas the primary oocyte of Triturus contains almost 600 nucleoli, and Xenopus, the toad, contains 1200 nucleoli in the nucleus of its primary oocyte. Now, another major change is the formation of informosomes. Informosomes were first reported by A.S. Sprin in 1964. What are informosomes? They are packed forms of mRNA protein complexes which are used for immediate production of proteins by the zygote, since the zygote cannot produce its own proteins immediately after formation. Now, the chromosomes often change to Lamb-Rush type. Now, if you remember, the Lamb-Rush chromosomes were first of all discovered by W. Fleming in 1882 in salamander eggs. Now, this change occurs during the diplotin phase of meiosis 1. Now, Lamb-Rush chromosomes are giant loop structures which help in increasing the transcription of mRNA and tRNA for rapid protein synthesis in the cytoplasm, and this happens by gene amplification. What does gene amplification imply here during oogenesis? It means that from now on only the oocyte selective genes will be expressed. The Lamb-Rush chromosomes are found in the eggs of some fishes, amphibians, reptiles, and birds, but they are not universal. They are absent in the eggs of sea urchins and mammals. There occur certain changes in the cytoplasm or ooplasm of the oocyte as well. The cytoplasm greatly increases in amount. The ribosomes increase in number for increased protein synthesis. The mitochondria also increase in number by autonomous replication. The endoplasmic reticulum, which are now known as the annulate lamellae, become more elaborate. That is, they now have more starch sheets with annuli or pores. This helps in transfer of contents of the nucleus into the cytoplasm, but however, these endoplasmic reticulum disappear in the late oogenesis. The next major thing that happens during previtellogenesis is the formation of cortical granules. Cortical granules are basically spherical membrane-bound vesicles which are formed from the rough endoplasmic reticulum and Golgi. Cortical granules are found just under the plasma membrane in the eggs of sea urchins, fishes, frogs, reptiles, and some mammals. Now, the cortical granules are filled with cortical enzymes and acid mucopolysaccharides. The enzymes consist of haline proteins, which help in keeping the blastomeres together. That is, they make the blastomeres sticky for adhesion after cleavage, so that they don't fall apart during the process of cleavage. The other enzymes are ovoperoxidase or the OVO, which hardens the fertilization membrane by cross-linking the tyrosine residues together and is spermicidal in nature. Now, these cortical granules, they block polyspermy by exploding at the site of sperm entry once a single sperm has entered the ooplasm. These cortical granules are formed in the endoplasm, but eventually they move to the peripheral region or cortex of the cytoplasm. And this cortex or the peripheral cytoplasmic layer of the oocyte, in which cortical granules are present, it's known as cortex. This cortex is actually semi-rigid and more viscous in nature because of the presence of these cortical granules in it. Now, this cortex also consists of actin microfilaments, which provide forces to help in cell constriction during cleavage processes, and also they alter the cell shape during gastrulation. From the cortex arise microvilli, which help in engulfing the sperm during fertilization. Now, this cortex region may also contain melanosomes, the pigment granules, which contain melanin pigment, and these melanosomes act as UV light filters. After the previtellogenesis phase is over, the growth phase continues with the process of laying down of yolk in the primary oocyte, and this phase is known as vitellogenesis, where vitellin means yolk and genesis means generation. Now, this vitellogenesis occurs in the egg when it is still arrested in the diplotin stage of meiosis 1. Here, the amount of yolk, as we see, varies from animal to animal. For example, in mammalian egg, there is just minimal amount of yolk present. Whereas in the eggs of birds, reptiles and sharks, they are quite large in size and thus they contain a large amount of yolk which is able to sustain the embryonic development to advanced stages also. Now, during vitellogenesis, there is this precursor of yolk which is first of all laid down, and this precursor is called vitellogenin. Vitellogenin is actually synthesized by mitochondrial aggregates called yolk nucleus of Galvani in the oocytes of spiders, amphibians, and birds. This process of formation of yolk can occur by two means. First is when vitellogenin or the yolk precursor is synthesized by the egg itself. So this method is known as autosynthesis. But when the yolk precursor is not laid by the egg itself, but actually by the extra ovarian tissues, then it is known as heterosynthesis. So these extra ovarian tissues or the tissues outside the ovary can be maternal liver as seen in vertebrates, it can be fat bodies and follicle cells as seen in insects. Now once synthesized in those extra ovarian tissues, the vitellogenin has to be carried by blood to the ovaries. And once it reaches the ovaries, this vitellogenin is taken up by the microvilli of the plasma membrane of the oocytes by the process of receptor-mediated endocytosis. There are some animals which may use both these methods, autosynthesis as well as heterosynthesis. So what is yolk? Yes, of course, yolk is the yellow portion of the hen egg that we generally eat. So what does it consist of? Yolk basically is the assembled stored food component. So it consists of proteins, phospholipids, neutral fats and glycogens which are essential as a nourishment for the growing embryo. And to provide this nourishment, the yolk breaks down into simpler substances. For example, the proteins will be broken down into amino acids. Now, yolk can be stored in the form of yolk platelets as seen in amphibian eggs and in eggs of some other vertebrates. It can be stored in a granular form which are known as yolk granules as seen in the case of invertebrates and some lower chordates, and yolk can be stored in the form of lipochondria which are essentially consisting of a core of accumulated lipid with an external protein cover. In vertebrate eggs, yolk is present in the form of platelets. So, yolk platelets are the large flattened ovoid membrane-bound organelles. They consist of two protein components. The first is phosphitin, which has got a molecular weight of 35,000. Phosphitin is a phosphoprotein in which more than 50% residues are phosphoserine. The second is lipovitelin. Lipovitelin has got a molecular weight of 400,000. It is a lipophosphoprotein which contains almost 17% lipid. Now, two molecules of phosphitin and one of lipovitelin form a clear hexagonal crystalline lattice-like structure, which is the structure of a yolk platelet. Now, this yolk platelets, they form around 45% weight of the amphibian egg and almost 90% weight of a bird egg. What controls the process of vitellogenesis? Predominantly, vitellogenesis is controlled by hormones which are produced during folliculogenesis along with nerve cells and follicular cells which grow in association with the oocyte in the ovary. Now, in insects, how vitellogenesis is controlled? See, there is an endocrine gland in the brain of insects known as corpora allatum or singular is corpus allatum, which generates juvenile hormone. Now, this juvenile hormone promotes the oocyte differentiation. It regulates the uptake of yolk proteins at the oocyte surface by the process of receptor-mediated endocytosis by the microvilli of the oocyte and also through the intercellular gaps of the follicle cells. It also stimulates the follicle cells to produce a hormone, ecdysone. Ecdysone, which is also known as the molting hormone, is converted to 20-hydroxyecdysone, and this is the activated form of ecdysone. Now this 20-hydroxyecdysone stimulate fat bodies present in insects to play. One in the process of vitellogenesis and second in molting, which we will study when we will discuss metamorphosis. And during metamorphosis, again we will study the same gland, corpora elytra, which helps in production of another hormone, that is prothoracicotropic hormone. So prothoracicotropic hormone as well as juvenile hormone both help in the production in the process of metamorphosis in insects. Now, two type of accessory cells play major role in extra-ovarian transport of yolk. As we discussed, the precursors of yolk are not synthesized in the ovary but in the extra-ovarian tissues. So, how and which cells play important roles in the transport of yolk precursors from outside the ovary to inside? They are predominantly the follicle cells and the nerve cells. In insects, as you see in the diagram here, there is a cluster of oocyte along with follicle cells, as you see here, plus nerve cells, which forms a collective egg chamber. Now, follicle cells are the ones which will uptake vitellogenin, which has been synthesized by extra-ovarian tissues and which comes via blood. And this uptake happens through the intercellular gaps present between the follicular cells and also through the microvilli by pinocytosis. Now, the other important cells here are the nerve cells. During oogenesis, some of the germline cells in the ovarioles develop into oocytes. What happens to the other cells? So, the other cells will become nerve cells or they will undergo atresia. So, nerve cells can be regarded as the abortive oocytes. That means those primordial germ cells which could not become ovum, they were aborted and thus they become nerve cells. Now, these nerve cells are polyploid in nature and their main function is to deliver large amounts of mRNA and proteins into the egg cell through microtubules that extend through their cytoplasmic bridges. Now, students, important to note here is that nerve cells sacrifice themselves for the process of vitellogenesis. Upon completion of vitellogenesis, the total cytoplasm of the nerve cells is engulfed by the oocyte. The process of vitellogenesis is very similar in insects and in vertebrates. So, in vertebrates, for example, in amphibians, it is controlled by the hormone estradiol. Just like in insects, there was this endocrine gland present in the brain named corpora elytra. In vertebrates, there is this hypothalamus present in the brain. Now, brain stimulates the hypothalamus to secrete gonadotrophin releasing hormones, which trigger the pituitary to release gonadotrophins. Now, these gonadotrophins in turn stimulate the follicle cells to produce estrogen. This estrogen helps in oocyte growth. It also acts on the liver to stimulate vitellogenesis. Interesting to note here that in different animal species, eggs are present in different stages of maturation at the time of sperm entry. For example, in Ascaris, Diacema, Crentia, Mesostoma, and in dogs and foxes, the egg is available at the primary oocyte stage. It is present in the metaphase 1 stage when released and available at the for sperm entry in case of Nemertean worm, polychaete worm, mollusk, dentalium, the core worm, pectinaria, in many insects including the starfish. Egg is present in the second metaphase stage in the lancet branchiostoma, in amphibians, in most of the mammals and fishes, as is also present in case of humans. Now, meiosis is complete in case of cnidarians, for example, the sea anemones and sea urchins at the time of ovulation and when the ovum is about to fuse with the sperm. So, students, this completes the lecture 1 of the module Gametogenesis. We have discussed what is gametogenesis, What are the different types of gametogenesis? We discussed both spermatogenesis and oogenesis. We have discussed the structure of sperm, the yolkling or the vitellogenesis process. Now in the next lecture, we will be discussing the structure of ovum and the regulation of gametogenesis in detail. Thank you. Hope you had a good time listening to me.